Welcome to Impact Quantum, the show that peeks under the hood of quantum computing to reveal what's emerging and why it matters. Today's episode is an absolute masterclass in quantum-inspired data science, with a guest who, quite frankly, makes the rest of us feel like we're still stuck figuring out long division. Joining Frank and Candice is Dr. Marvin Weinstein, emeritus at Stanford University, bona fide particle physicist, and co-creator of dynamic quantum clustering, DQC, a method that sounds like science fiction but delivers real-world, potentially life-saving insight. Marvin takes us on a thrilling journey through brain cancer research, data agnosticism, and how a physicist wandered into biology and found patterns that even seasoned researchers had missed. This isn't quantum computing per se, it's quantum mechanics-inspired analysis applied with surgical precision, minus the surgical gloves. Whether you're a curious technologist or just here for the intellectual thrill ride, this one is for you. And no, you don't need a PhD to follow along, just curiosity and perhaps a cup of strong tea. This episode is rated 5 Schrödingers. So buckle up and let's get into it.
Hello and welcome back to Impact Quantum, the podcast where we explore the emergent fields of quantum computing and the upcoming ecosystem that is going to sprout around it. So you don't need to be a quantum physicist, but you do need to be curious and curious about quantum computing. And with me today, as always, is the most quantum curious person I know, Candice Kahuli. How's it going, Candice?
It's great. Thank you so much for asking. I'm really excited about today.
Yeah, so I think today you actually have an honest-to-goodness physicist here on as a guest.
Absolutely. Amongst many things that he's doing, I can say that he is a particle physicist at Stanford University, as well as the CSO co-founder at Quantum Insights Incorporated. He's got a lot of experience and a lot of great knowledge to share to our audience. I think everyone's going to find him as fascinating as I do.
Cool.
I hope. But I am a genuine quantum mechanic. That's right.
There you go. So please welcome everybody, Marvin Weinstein to the show. How's it going?
It's going well. As I was telling Candice, you got me in a very excited state today. So I hope I'm coherent.
Awesome. Yeah, you in the virtual green room, you had said you kind of uncovered something very interesting. So we can start there if you like.
Well, yeah. I mean, basically, the thing we were talking about during the previous interview was a tool that was developed. That was what the company was founded for, to apply to various problems. And it's called dynamic quantum clustering. And it differs from other clustering algorithms, other data mining tools, in that it is completely unbiased. You can take a first look at data with making no assumptions about if there is anything to be found in the data, cleaning the data, or labeling it in any manner, shape, or form. You just look at the raw data. So for personal history reasons, I mean, Candice was telling me about somebody she knew who partner had died of, or father had died of a brain tumor. But my first wife also died of glioblastoma. So when Quantum Insights decided to close its doors, I was sitting with all of this data from the Cancer Genome Atlas, including all of its glioma data. That means all low-grade gliomas and glioblastoma data. So I had RNA sequencing data for all of those tumors. And basically, I decided first thing I want to do with it is take a look at it and see if there's anything to see in that data. That, I mean, people have looked, this data set's old, so it's been around for a long time. It's been heavily studied. People were totally sure that everything that there was to be extracted from that data set had been extracted from that data set. And so basically, I said, Well, nobody's looked with our tool. And so what did I do? Well, the first thing was, as I promised you, I simply loaded up the data. I did restrict the gene expression from the 60,000 genes that it comes with down to what everybody believes is the 20,000 so-called protein coding genes. Not all of them code for proteins, but they're the list that various tools restrict to. So I wanted to stay within what other people were doing. So I looked at those 20,000 genes. Well, that's a lot of data. I mean, that's a lot of noise. It's actually not a lot of data. It's only 600. I mean, that's always a misconception. People say biologists have huge data sets. They really don't. I mean, for example, all of the cancer data is 692 tumors, brain cancers. That's not a big data set. There's only 692 pieces of information. I don't care that there's 20,000 genes, because all you're seeing is the effect of 692 combinations of the expression levels for those genes. The whole data set can be reproduced from those 692 pieces of information. So, not like a physics data set, which has millions of samples and stuff to look at. This is a biology data set, and typically restricted to a specific disease. It's not huge. What it is complicated and really hard to see what's going on because there's so much noise. So the first thing I did, as I said, was restricted the raw data. It's a matrix after all, rows and columns. Okay, every row is the expression level for 20,000 genes. I'm rounding the numbers off. You don't want the 20,312 all the time. And so it's that, and there's 692 rows in all. You feed that into DQC, it's made to ingest that quickly. And you just simply run the first analysis. And surprise, the first thing you see is a whopping big signal. In fact, that data, raw, unprocessed, unlabeled, untreated in any way, no training set, separates into two clusters. One very large cluster, which is mostly the lower grade gliomas, and another cluster, which is almost all of the glioblastomas. Well, that's pretty cool. There's already a signal. It's not the best classification in the world, maybe. It's very good. It's competitive. But DQC has a standard trick, which is you can pick out a smaller number of genes to look at, in this case, a smaller number of features in the fancy language, which give the same information. And so first run at that produced 544 genes and exactly the same picture. So I didn't have to look at 20,000. I had to look at 544, which were doing most of the heavy lifting. Produced the same two clusters, same, not wonderful, but pretty good classification scheme. And then there's another DQC-based trick, which is using the information in the two clusters now. I can order the genes that I'm looking at, the 544, in order of their importance to the signal. So then I look the first 10 genes, the first 20 genes, the first 30 genes, and I did those analyses over and over. Each time I did it, starting from 10, I got a pretty good classifications. 20 made it better, 30 made it better, until I got up to 90 genes. And then at 100, 110, 120, everything stopped getting better and started to get worse. So the cutoff point was I wanted to look at the 90 gene signal because the cleanest information was going to be in the 90 gene signal. Did that, and sure enough, I find 4 clusters. So what are the 4 clusters? 3 of those clusters are all low-grade gliomas, 100% low-grade gliomas. They capture all of the low-grade gliomas except for 4 tumors. The 4th cluster is all of the glioblastomas and those 4 that were not captured. Now, remember, there were 692 tumors. I was missing 4. So when you look at them plotted in the space, let's call it PCA space. You like that word? And it is the PCA space for the tumor expressions. Those four lie right next to the gliomas, whereas all the other data lie far away from the gliomas.
And
So
just for folks that may not know what PCA is, is it principal component analysis?
Principal component. Yeah,
And it's
PCA is a way of rotating the data so that the dimension of the data in which the data is most spread out is the first dimension. The dimension in which the data is next most spread out is the second. It tends, if you're lucky, in low dimensions to show you what you need to see in order to try to do clustering, because most clustering algorithms deteriorate rapidly as the dimension of the data goes up. So they like to do a hard dimensional reduction, they call it, to 2 or 3 PCA directions, and then try to cluster based on what they see there. There are algorithms which work in higher dimension, but still, there are things they struggle with. DTC doesn't really care. It doesn't start with a hard dimensional reduction. It simply works with what is showing the most information. If it's 6, if it's 10, if it's 16, if it's 50, that's fine. I don't care. I'll work in that. The only impact and price I pay is the time it takes to run the algorithm. And so the usual trick is you work in the lowest number of dimensions that appear to be noise-free, which you can tell by looking at the spectrum that you see in PCA, and then work your way up to twice that number of dimensions and look again. And if you see the same information, well, it's quicker to run everything in the lower dimension. But you don't stop if you see a change. So at any rate, ran it. I got these 4 clusters. Now you notice the only misclassification out of 692 tumors is 4 tumors. So a considerably less than 1% failure of doing it.
Close to like 1/7 of 1%, would you say?
Yeah.
So I mean that's
I'm trying to quote the worst possible statistic that I can imagine.
Fair enough. But less than 1% we can all agree on. So that would put it at over 99%.
At any rate, if I tell you from this analysis you have a low-grade glioma, I'm 100% accurate.
Right.
If I tell you you have a glioblastoma, I might be as much as 2.5% inaccurate on just glioma question. That's better than world class.
I was going to say, what's current state of the art? Is closer to like 80-20,
I've been looking
90-10?
around trying to say, how do we compete? And I haven't succeeded yet. My collaborators, one bioinformaticist at Wisconsin and a cancer doc at Stanford, are going to have to help me with that in searching the literature. What I find are statements like most schemes for doing this unsupervised from the raw data and then moving on from an internal analysis, still working, starting with the raw data, what they call the area under the curve. So the likelihood you're right is 70 to 80%.
Interesting.
So we're not talking anything like the same. There are some special biomarkers. If they're found on a glioblastoma, then people are pretty sure it's a glioblastoma at maybe the 1% level. Okay, but separating glioblastoma from low-grade gliomas blind, they're nowhere near that good. At any rate, that's what I found. I now have the world's best classifier in 90 genes. I plot the gene expression levels for each one of those clusters. And for most of the genes, I see the genes either fall into the category, the expectation value for the expression of that gene either goes systematically up through the 4 clusters, going from the lowest grade glioma to the glioblastoma, or systematically goes down. That's what you want to see. Those genes are involved in what's happening. But there's still 544 genes, and I can't see the forest for the trees.
Interesting. So does this inform treatment options?
Well, that's the problem. Treatment options, or at least my understanding. So remember, I have to be very upfront. I'm not a biologist.
Right.
Everything you will hear me talk about, I learned by looking at this data. I have no formal training in biology whatsoever. So you're dealing with a novice.
It
Okay.
reminds me of the original Star Trek show where Bones would always like, "I'm a doctor, not an engineer." Like you're like, "I'm a physicist, not an engineer." I mean, a doctor. You know.
So
That's right.
what initially inspired you to take all of your quantum mechanics knowledge and
Oh,
apply it
that's
to
a
biological
that's a
data?
yeah. So remember, I'm the co-inventor of this algorithm. The other inventor is David Horn, Tel Aviv University, a frequent visitor to SLAC. He collaborated on many physics papers.
And
Uh,
Slack is not the
the
messaging app. It's Stanford Linear Accelerator. Is that?
Right, no case. Stanford, it used to be called the Stanford Linear Accelerator Center.
Mhm.
So I'll tell you out of school a story, which reflects wonderfully on the DOE. At some point, the DOE wanted to put its name on everything and trademark it.
Mhm.
Well, Slack said, because Stanford said, you can't trademark the Stanford Linear Accelerator Center. It's us.
Right.
We run the place. So DOE made Slack change its name to Slack, SLAC, and call it the SLAC National Accelerator Laboratory. So I guess as an abbreviation, we're now SNAL, not SLAC. SLAC sounds better. I grew up with it as SLAC for 42 years. The hell with the DOE. I don't intend to listen to what they want. But it is now officially the SLAC National Accelerator Laboratory.
Right.
So at any rate, David Horn came into my office and life went as normal. He said, "Oh, I have something interesting to show you," because he kind of had left high energy physics about 8 years earlier and was looking into data mining. And he said, there's this cool idea that grows out of something done by somebody called Emmanuel Parsons, called the Parsons estimator. And I figured out I should think about it as a quantum potential. I already was very suspicious. It sounded like a very strange idea. And so we did our usual thing. We stood at the blackboard and yelled at one another for 3 or 4 hours. And then we came to a meeting of the minds and said, this really isn't a stupid idea. It's kind of cute. And, David said, well, he showed me some simple problems having to do with classifying crabs. It's the standard old problem that people did. And seemed to be very interesting. He said, but the problem is, in order to understand what the idea behind it is very simple. You take all the data, you create a function. The properties of this function are wherever there's more data than it is in a surrounding, there should be a peak. And wherever there's less data, there should be a valley. The problem is, of course, the way you create that data is very sensitive to a parameter that you introduce. Okay, I don't want to get too messy in this. It's all published, so it can be read. And the sensitivity is hard to deal with. So what we finally understood was if we treated this, so this is just a professional deformation. Because we're particle physicists and quantum mechanics, we think of everything as having something to do with particle physics and quantum mechanics. This problem has nothing to do with particle physics or quantum mechanics, except we want to get rid of the sensitivity of that function. And we said, if you think of this as the solution to a problem in quantum mechanics, that problem has a term having to do with the particles moving around, and another one having to do with the landscape it finds itself in. That's called the potential function. It turns out that potential function always has sharper features, more pronounced dips than the solution has peaks, and turns out to be much less sensitive to the parameter that goes into building that function. So literally by saying, what problem is this picture the solution to, which turns out to be trivial to solve, What potential function? You get a sharper picture, and the sensitivity, the parameter used to build what's called the kernel function, that potential function, goes down by a factor of 10. So you have a pretty unique answer. It's easy to arrive at. You don't have to be careful about picking your parameters. The problem is, if you think of this as things living, so we have these valleys now where the bulk of the heavy concentration of data is, or we have stream beds. But the data is up along the walls as well as being down in the valley. So the question is, which data belongs to which valley, which stream bed, etc. And so you want to move the points down the sides of the valley and have them collect in whatever structure is at the bottom. Well, people try that. In fact, my colleague had been trying it. And as the dimension goes up, for reasons we understand, that surface becomes rippled just due to noise. And so basically, if you try to just move things down using ordinary calculus, what's called gradient descent, you're just moving points in the direction of the slope, they get stuck in the ripples.
Oh, I see, because it can't find the global minimum.
It can't find the important minimum.
Right.
If
Now,
things move according to quantum mechanics, all bets are off. It's a much nicer story. And it's the uncertainty principle which made the solution wider to begin with. So we're going to exploit the uncertainty principle. If I move points according to the laws of quantum mechanics, the first thing is, unlike gradient descent, the quantum wave function extends out to where the valley starts going up again. So points automatically start to slow down as they reach the minimum. And they don't overshoot and rattle around, they just stop. Because they see now equal influence from both walls, and therefore no force.
right.
Also, they don't see ripples because the uncertainty principle allows for quantum tunneling, and they simply go through those tiny ripples or right above them. So as a way of making the data move and find the minima in the function in any number of dimensions, and there's a way of speeding up the analysis because quantum evolution is done by matrix multiplication, so it's enormously parallelizable. Didn't say that very well. Parallelizable. You get a very quick algorithm that is using physics principles, but to solve a non-physics problem, just getting the points efficiently down to the bottom. If there is a riverbed, that tells you something about the data. It says there's some one parameter thing, some regression on the data that you can do to something that's very extended. It's a huge discovery. It's much better than finding simple clusters, huh? But that's what this does. So DQC has advantages. One, it doesn't require training sets. So it's great for biology data because having annotated training sets that are really good, hard to come by.
So this is interesting. And what does DQC stand for?
Dynamic quantum clustering, meaning that we're
Dynamic
using quantum mechanics to find the minimum.
Now, do you need a quantum computer to do this, or this is just an algorithm?
I
Interesting.
told you I'm here under false pretenses. You asked me here to talk about quantum computing, and I told you I don't do quantum computing. I'm talking about using quantum mechanics to run on an ordinary computer. Could it run on a quantum computer? Yes. If they were really as fast and as good as they say they're going to be, would even be better because it can handle bigger. But I'm focusing on biology, by the way. This algorithm is data agnostic, right? It's not talking about biology per se. It doesn't care. It just says that there's something interesting. Data is not distributed with equal density every place. Things that are more like one another tend to be located in a more dense region. Okay, so, and this has been applied to many things. It's been applied to finding radioactive sources in the city of Chicago hidden in a building. Okay, there's a paper that I wrote on that. It's been applied to, I guess there's no paper on this, but it was a problem I did for somebody. Finding tanks in the desert that have been camouflaged, painted, same thing, using the data from a multispectral, hyperspectral camera, they're called. So it doesn't care what the data is. It's data agnostic. It's feature agnostic. It is unsupervised completely. That doesn't mean that you don't use the results of a previous analysis to now supervise the next analysis based on what you learned. You do that. But at any rate, that was it. So what's now going on is, as I said, we have the world's best classifier, but I don't know how to tell you what the best drug for your tumor, the one that's most likely to work on the biology that's happening now, should be. And that's why I need to go find a biologist. And they're not so great at doing it either. So witness how many people go through many, many failed drugs.
Yeah, and
Well,
precision medicine is definitely, one of the biggest outcomes of using this type of clustering that we can create. I mean, there's so much out there that needs this type of, this type of
it's still in its
viewpoint.
infancy. It's got a
Right.
ways to go to be precision medicine.
Where do you
At any
think?
rate,
Oh, go ahead.
oh, please don't let me. What did you have to say?
I was going to say, based on dynamic clustering, quantum clustering, where do you see it evolving in the next 5 to 10
I'll finish
years?
telling you this story about why I'm excited, because I think it's evolving to a really. I've seen something today I never thought I would see. So last night it showed up at 10 o'clock in the evening, and I'm still digesting what I saw. So what I show you should take with a grain of salt, but there's no question, there's zero chance that I'm wrong in terms of what you'll see, okay? So the way docs like to look at the problem, or cancer researchers, is they talk about so-called biological pathways. Biological pathways are sets of genes which carry out some process. In the end, all processes are making proteins, but we're not looking at the proteins being made. But we know these sets of genes are functioning together to produce an interesting output. So if I can take the information I have and find a way of saying, oh, so in fact, what I'm seeing is actually predicted by the following set of genes, and I can assign meaningful coordinates to each tumor based on where they are in what that set of genes is doing together. I'm in biospace. A point in that space, depending on how many things, still I'm producing one axis in biospace. And it's representing a process which is happening in the patient, where a bunch of genes are telling me something, not one. And that bunch of genes I can look at and ask, what are their properties? What are their common properties? So I will share something with you. So at any rate, did that, okay? Went to biospace using DQC methods again. Remember, I told you I had 4 clusters. So there's 6 pairs of clusters, which differ in how the genes are being expressed in those clusters. So I can find the list of the most important ones between 1 and 2, 1 and 3, 1 and 4, 2 and 3, 2 and 4, 3 and 4. So 6 possible axes in biospace, the sets of genes that are most important. And then using those axes, which go from minus something to plus something, I can assign a coordinate to every one of the tumors. So I have points in a 6-dimensional space. Okay? The way that's done, it's done in a way such that 0 on that axis means that for that set of genes, that point is consistent with what the value for all of the genes in that thing, the average value of those genes, is. Plus means you are moving X standard deviations away from being at the average expression. So I don't need to know what the normal expression of a gene is. That's always one of the problems. You rarely have data for normal cells of the same type as the tumor. And so you don't know where to set your zero. Here I'm doing it by the average, and I'm saying how far from a standard deviation am I out one way and how far out am I the other way. And so you plot the same tumors. Now I have to remember what I do. I go to share.
Yep.
Share the screen. So you plot the same set of tumors. Now you see my background.
Yes.
And I am going to switch over to the computer in my basement and show you a fun thing. So the axes you see here are DQC's plotting of the cancers in a six-dimensional biospace. What I want you to see, blues are glioblastomas, reds are the lowest grade gliomas, magentas are the next lowest grade gliomas, and the golds are closest to the glioblastomas.
Interesting.
Now, I told you this is an animation. We're going to start the points. This is how DQC works, okay? So we're moving the points downhill. You like that?
Yeah.
So it's all converging.
What's happening?
They're all
Not only
converging
that,
into
this
one.
is like a regression, right?
Right.
It's a one-dimensional shape. The healthiest tumors, they're not healthy, but they're the healthiest.
They're not the least awful.
And yeah, the least awful. So if I look at for this, we already saw that when we analyzed the, so the colors here are the clusters that I discovered in RNA sequencing space, in what we call gene space. They've just been arranged in a line from best to worst.
Oh, so blue is the worst?
The glioblastomas over here.
Okay.
Mm-hmm.
Okay.
So for those listening, don't worry. We're going to link in the show notes to a video representation of this. Interesting.
At any rate, this is what you see. So this is the plot in biospace. Now that's very interesting because these have the dimensions of the bio coordinates, and those coordinates have a meaning. Okay. In fact, I'll tell you what the meaning is.
And this is based upon a set of data of patients.
Yes, this is 692 patients. That data was submitted to the Cancer Genome Project.
Okay.
Oh, so this is open source data that you're pulling in.
This is absolutely open source. What
Got it.
my company did when we existed, because we had various projects going and things like this, we downloaded all of that data for the RNA sequencing data, and as much as we could find about each of those tumors, which was not a hell of a lot, but there's something. It's a good database. As I said, it's been studied for years and years and years. So this is results obtained by starting from no information and just re-looking at the brain cancer data and saying, people have been studying this forever. Did they ever find anything like this? And the answer is no. This has never been discovered. This has never been discussed.
So using traditional analytical sources, you could not
Clustering,
get
whatever.
you could not get at this information without doing the dynamic
right,
quantum
because
clustering.
you make a lot of assumptions about what you're supposed to look at. You make a lot of assumptions about how you filter the data. You end up throwing the baby out with the bathwater. Having
And you
something
also
that's go ahead.
No, you also said you didn't do any cleanup of the data. Like that's just the raw.
No, well, I mean, they've cleaned it up, obviously,
Right,
at some
right.
level. But we're not doing the post, whatever they did, cleanup that people normally do, where they filter out genes where they have, this gene should be expressed at least at this level. All genes that aren't expressed at that level, we're throwing out of the data set. Okay? If I see a difference between two clusters and the genes are expressed differently in the two clusters, but what they call the fold value isn't big enough, I'm throwing it out of the data. Well, you can imagine if there's hidden information in the data and you're busy throwing things away the chance you throw the baby out with the bathwater is very high. Okay?
Exactly.
And that's exactly what this shows. The benefit of going in unbiased, unfiltered, completely agnostic look to see if there's a signal first. And then when you see the signal, which I did, so stage one is, wow, there's a signal. Stage two, what is making the signal? EQC is built for solving those problems.
Right.
So basically, and that's where it differs from AI, okay? AI needs training sets for the most part. There are versions of AI now that claim not to, which are real. They make up data in order to train the data. There aren't enough training sets. So what you do instead is you make up artificial data and then try to teach it to reconstruct the real data, okay, by picking the parameters in the artificial data. And then you try to classify existing data. But it's a different story. Here, everything is understood. The algorithm is totally prescriptive. I know exactly what's going on. There's no mystery once I find something. And we ended up, I just showed you, with this concept of biospace, which is what people in the literature, it turns out, that's where the idea came from, to look at it this way, what people were talking about as latent coordinates in the data. So there are people doing AI that say, oh, I'm going to keep feeding AI from this, and AI is going to reduce my problem to some low-dimensional manifold, and I'll call that a latent coordinate picture. But then I'm faced with the problem. I don't really know what the coordinates mean. I'm busy trying to interpret them, and I certainly don't know how to exploit them. Different here, right? So we started with no training data. What am I looking at here? Shouldn't be showing you this, but my collaborators say I can show it to you. So here are the axes. So what do you know from these axes? Well, the genes in this axis have, as they say, tumor-associated fibroblast activation. They're immune checkpoint genes signaling chemokine-driven inflation. The pathways that are being recruited for this or that. Basically, here it says, this, if you want to change overexpression or underexpression, you want to look at the drugs which do the following thing. There's one such description for every one of the 6 axes. They have a meaning. And so if I simply look at the coordinates in biospace and see along which of these axes the biggest signal lies, that's the first set of drugs you try on the tumor. So by looking at it in biospace and how the tumor evolves in biospace, that's what this is, right? The evolution of the tumor in biospace. Every one of these points, after all, is a snapshot in time of a tumor at that point. What this suggests is it's a continuous evolution to glioblastoma through these biological processes. And as they change, so you're seeing so basically, what have I learned? God is showing me, or biology is showing me, how the tumors evolve in time.
Interesting.
I don't know. That doesn't.
So do they all start out as like, if you showed that image again, but the one where they're all on the same plane? The one that we're all on the same plane.
This is the snapshot in time. This is the snapshot in survival term for the patient. Okay. Because that's what is changing along this curve. We already saw that.
Oh, I see. Okay.
So this had different survival times. So these are all tumors. I don't know where healthy is.
Okay, but so not everybody starts out, for example, in the red, and then based upon
they probably do.
Okay.
Most glioblastomas have to be, if you look at them in terms of their gene expression patterns, they're a mess. Okay, they've undergone many mutations to get where they are.
And the more mutations, that's the different colors, basically, and they change. Okay,
right?
so everyone starts out maybe with the red, but not everybody goes all the way to the blue, purple.
Right.
And they
Everybody
probably gets
probably starts
worse.
out to the left of the red
Right?
because these tumors probably form at the single cell or small number of cell levels
Okay.
and take 10 years to grow
Okay.
to
Oh,
first show
wow.
up and be seen. Okay. So it's not, we don't have examples of the earliest version. That's the beauty of what's blowing me away.
Yeah.
Don't need to know any of this. I don't have to know. I only need the gene expression pattern. And I only needed the information about survival time to interpret the axis. Everything else came after I found the axes when I had to interrogate pathway databases to find out what they do. And truth be told, I asked an AI to give me the information about that because it's a pain in the ass to go through those things yourself.
So we could
So, I mean,
use
and I just wanted to know what I might see. This is not to be
Right.
taken seriously, okay? Because my biologist and my doctor friend are going to have to do the job of vetting what these interpretations. I only trust AIs a little bit, but it's sort of fun to do that, okay?
Right.
But what I wanted to give you was a feeling with the difference between biospace information and simple single gene information. Okay. And it's awesome what the difference is. And it's awesome that there's a progression in biological processes that lead you to glioblastoma. I can't tell you this actually represents evolution. But if it looks like evolution and it smells like evolution and it quacks like evolution, it's evolution. Okay. I mean, that's just my feeling. Now, I've already given you all the, I don't know any biology, do know a lot of physics, do know how DQC works. Okay. I know that better than anybody. But this business that you can take the information that you learned in the genetic, right, in the single gene basis, and convert it to biological process basis and learn entirely new things, more suited to advising doctors who are treating cancer patients, because I can take a new tumor stuff and put it on that plot, see where it is, see what its axis definition is, and see what the likely best drug is to start with. And then if that doesn't work, drop down to the next most likely, the next most likely. So basically, that's sort of, we can stop sharing actually now, which he says I can stop sharing.
There you
Okay,
go.
great. So you know why I'm in this befuddled state at the moment? Because I am still absorbing what this is telling me. I certainly never expected. When I thought of trying that, because people talked about these latent variables and hidden dimension, hidden coordinates, and describe ways it might work. I didn't see any examples actually worked out. This is the story from beginning to end, from
Right.
genetic coordinates to discovery to the world's best classifier to changing that into bio coordinates discovered from the genetic side. The treatment options, a tool for helping doctors treat, for suggesting to cancer researchers new experiments to do to verify what they're seeing on this. There's lots of suggested. I alone, with no knowledge, can think of 10 things people should explore based on this. And drug companies want to know what the next set of things to target should be for a given disease. Wow. I think that's pretty cool.
That is impressive.
So it really is, DQC is telling me the data is whispering to you. I'm the tool that'll teach you how to listen. That's the way I feel
That's
about it.
cool.
Since it's my baby, it's grown up. I really think it's grown up. And I'm very impressed with where it got. So you're getting me in my very biased statement form at the
Oh, I
moment.
can tell. It's super, super humble. But no, it's really exciting. But also to see where it can be taken from there.
Oh,
Like this is just the
absolutely. I
beginning.
mean,
There's so
this is
many.
scratching the surface. First place, those
Exactly.
axes could be improved because there's more than one set of genes that give similar information. Um, how to exploit it, how to do the bench experiments, that's not me. I don't know that stuff. And I'm 83. I'm not ready to start learning how to be a bench biologist. Okay. But it's just so cool. I mean, it's like you've seen the underbelly of what's happening in the biology. At any rate, I don't know if you agree with me, but I think it's really cool.
No, that is really cool. There's a lot to take in.
Yeah,
Lot
I'm
of
sorry about that.
no. I mean, we have a scale system for these shows, right? Like
What is a what is it, a five Schrodinger, you said?
Schrodinger's, yeah. So we have like from 0 to 5 Schrodinger's, and this is definitely going to be a 5 Schrodinger show. Like, and I was able to follow on because I was a data scientist, before this. So like when you said PCA, like I knew what you were referring to. At least but like so like it was like this show is really geared towards the quantum curious. Some of which will be data scientists, some of these will be marketers, some of those will be, kind of traditional software engineers, et cetera, marketers, right? Because it's our thesis that when the quantum computing ecosystem comes around, and indeed, I think what you've proven today is you don't really need quantum computing to take advantage of the innovations in quantum science, right? Like,
Right.
I think that was an assumption I think Candace and I had. I don't want to speak for Candace, but I know I certainly did. But I know that there is a field called quantum-inspired algorithms, which is probably what
That's sort of
you're.
what this falls
Yeah.
into. Yeah.
But it's just exciting that innovation like this can come about in such a way that it's going to improve people's lives. What you've discovered is, I'm not a biologist or a doctor, but I would imagine that a doctor or pharmaceutical researcher would look at that and say, oh, you know what this means? This means X, Y,
I
Z.
hope so. I mean, I'm pretty much at the limit of what I can do, even with collaborators on our own. The point we're writing the paper now, I've already blown my collaborators out of the water because this was discovered last night and they don't know about it yet. I have their permission to talk about it though. So
That's cool.
it's you know, so I'm glad you liked it in the 5 Schrodinger level because I'm only here because you guys refused to accept my statement. I don't know anything about quantum. Well, I do know something about quantum computing, but I am not a quantum computer person, and so I didn't belong on your show. But you kept refusing to let me off. So
But I mean, I think it's important that people think about like this is not. I think one of the things that obviously you're a great presenter and great teacher of these very complicated topics, but you've also something figured out. Plus, I also think it's important for people to realize that quantum physics and research in that space is already improving people's lives, or at least already showing fruits of that. Um, and I think that your research kind of shows that. It's like, you don't have you don't have the billionaires facing off over, Jensen saying it's going to take 20 years, Bill Gates saying it's going to
this
take
was low
less.
budget.
Right. I mean,
The
this is
machine's in my
pretty.
basement, and the data is free. So,
There you
well, I
go.
think the other lesson here is we have a wealth of data that's underexplored because looking at it in an unbiased fashion hasn't been done.
Right.
So I have lots more diseases I want to look at. I have all this TCGA data for pancreatic cancer and various other cancers. And so it's sort of fun.
Right.
I like how you kind of mix, I know you say you weren't appropriate, but I think you were totally appropriate for the show. And you've got the physics background. You're talking about quantum clustering, why it's affecting the biological, giving us biological data that we're able to move forward with potentially for precision medicine. I love the bridges that are being created all over the place here, that you're not just kind of stuck in one thing, thinking you can only do one thing because you have a certain amount of knowledge, but how you've bridged that to bring in all of this biological data information. I think it's fantastic. I'm very happy that you came and you joined us today. I learned
Well, I'm
a
glad I
lot.
didn't bore you, and I hope I didn't get too far into the weeds, which my wife accuses me of doing all the time.
Ah, mine too.
Yeah.
Where can people find out more about you and what you're up to?
Me and what I'm up to. Well, I'm
Yeah.
on LinkedIn. People
Okay.
contact me through LinkedIn all the time. Uh, I have a long history, and if you go look at the archives, the physics archives, PhysRev A, PhysRev B. I mean, my past history is a little eclectic, even in physics, which I attribute to having a short attention span. But I started in particle physics in phenomenology, it means looking at data, trying to understand what it's telling me. I moved into pure abstract particle physics, and then I went into what's called lattice field theory and lattice gauge theory, which is trying to learn stuff from how to say this. Didn't expect to talk about this. So let's talk about how we do physics, which is another totally off-topic thing, and you may be running out of time. I don't know. You tell me when I have to shut up. But the story is, physicists are smart, but there are very few problems we know how to solve exactly. Only a handful. Everything else is done by a process we call perturbation theory. Mathematicians also call it perturbation theory. You say, well, this problem that I know how to solve exactly kind of looks a little bit like this other problem, but with, say, some modifications. So let me add the modifications to the problem and try to calculate corrections to the answer based on the modifications. So I have the original problem set and forces involved and the changes in those forces a little bit. And then I calculate perturbatively what's happening. People do it in celestial physics all the time. I have this planet moving around the sun in an elliptical orbit. Oh, well, but there's the moon. So how does that affect the orbit? Well, I can't solve that problem. That's already a three-body problem, and there's
Right.
no exact solution to the three-body problem. By the time it's also got Mars and Jupiter and Saturn and Pluto and Mercury in the problem, I can't plot orbits.
Right.
But people do it all the time. NASA plots orbits. How do they do it? They calculate the original orbits, and they start calculating the effects of Mars and this and that on that orbit, because we know what those forces are if Mars is on its orbit. And through successive corrections, successive iterations, you're able to make the small perturbations in the orbit that get the answer right for you, and eventually let you send something to the moon and not miss. Okay. So perturbation theory is what we use. But what is perturbation theory based on? I have a solution I know how to get exactly, and I know how to make small corrections to that solution. And then I can describe all kinds of crap. So, for example, condensed matter physics talks about matter. So I ask you, has anybody ever proved that the table you're sitting at exists? Is there such a thing as a table made out of wood? In fact, is there such a thing as wood? The answer is no.
Right.
Use wood to build houses. I use engineering principles to calculate the stress load on a beam. How the hell do I do that if I don't know wood exists? I describe wood. I assume it exists. I characterize it in terms of a bunch of properties. And then I can, based on that, make small correction calculations again to see how the wood behaves when I stand on it. But I have to start from the assumption it exists and that there are properties I can measure for it and make predictions based on that. But the first principles thing that would exist, no way, nobody solved that problem. Okay, so I was very interested in that because that's sort of a first principles problem.
Right. It's
And
very philosophical, isn't it? It's where a hard science like physics kind of meets up against a
oh, we
very
meet up
soft.
against soft stuff all the time, and
Right.
we fail to solve the problem. But that's okay. It's at any rate, um, I was always interested, always. After many years in phenomenology, and papers published in phenomenology and things like that, uh, getting into field theory and trying to understand from first principles how to solve hard problems. That, like quantum chromodynamics, that intrigued me because we're kind of using up this perturbation theory paradigm. Okay, it's very useful, it's very good, but we're already running into lots of problems where it doesn't work. We don't know a problem that's approximately like the problem we want to solve. So
Right.
how do you solve it? So I got involved in that. I got involved in what's called lattice field theory. And then I said, but how am I going to know I'm right? Because I could be wrong in pushing my answer in the one known direction. There got to be other problems, but there's only one quantum chromodynamics. It's the one we live with. It's the one we're made of. So I don't know if I'm cheating or not, but there's lots of condensed matter problems, and they all
Right.
have different answers, and many of them are strong coupling problems, and you can't treat them perturbatively. So take the same methods and change your field and go look at condensed matter and see if you can develop techniques to do that. Then did that for a long time, and then developed some methods and decided, oh, David Horn came into my office and I said, oh, this looks interesting. So I can't stay in one area. Now, to me, it makes sense why I'm changing. To other people, it looks like I have no attention span. So that's okay, because I do this for me. And so as long as I see the thread, I'm happy. But that's how I'm here. I'm now in biology, quote. But
Well, we're glad. We're glad that you're here. We're glad that we got to
Yeah.
learn a bunch of stuff today. I think it's going to be really exciting to unpack it, and to have you back, because you are just a
well, it's fun
wealth.
talking to you guys, but I'm going to bore you soon.
No, I don't feel
Most people,
bored. I mean, I'm more fascinated. I'm confused. It's about some things, but like I'm also fascinated too. And we want to be respectful of your time, and but
well,
we'd
I
love to
told
have you
you, I'm
back on the
retired.
show.
I'm sitting in my office. I have
Well, then
nothing
we'll
on
definitely
until
have you come
5 o'clock
back.
this evening. Huh?
Awesome.
We'll definitely have you come back then, because again, it's just really great information. It's important. It's exciting. I think it's very exciting.
Yeah.
So
Uh,
unfortunately, we have a little limitation. So
yeah. But definitely, and so folks can reach out to you on LinkedIn and engage with you directly if you're cool with that. And we'll let our
Sure.
AI.
I don't promise to answer everybody.
Right.
And if they're a crackpot, I don't promise to be polite.
there you go.
That's fair.
I'm
That's
liking that.
100% fair.
I like that.
We'll let our AI finish the show.
And that wraps this Quantum Odyssey on Impact Quantum. A massive thank you to Dr. Marvin Weinstein for taking us deep into the fractal jungle of biology, data science, and quantum mechanics with only his brain, DQC, and a suspiciously underutilized basement server farm. From classifying glioblastomas with 99% accuracy to uncovering biocoordinates that could revolutionize precision medicine. Marvin reminded us that sometimes the biggest scientific breakthroughs don't require a billion-dollar lab, just a stubborn physicist, open-source data, and the audacity to ask, what if? If you enjoyed this episode, and really, how could you not? Be sure to subscribe, share, and let your fellow quantum-curious friends know. And as always, check the show notes for links to Marvin's work, ways to connect, and possibly a diagram that will make your head spin just a little less. Until next time, stay curious, stay entangled. And remember, just because you can't observe the quantum doesn't mean it's not observing you. Cheers.